Hi everyone, I'm Nico and today I'm going to present my semester project on ACD as a target inhibitor for metabolic regulation in breast cancer, specifically in triple negative breast cancer cells, or what they are commonly known as TNVC cells. The reason why I chose ACD as my target drug is because it targets a critical metabolic vulnerability in TNVC cells. These cells depend heavily on altered energy pathways to grow and survive. Unlike other breast cancer subtypes, TNVC lacks hormone receptors, which makes it resistant to many standard therapies. And it also makes them one of the most aggressive types of cancer to treat. ACD targets two monocarboxylate transporters, MCTs, in specifically MCT1 and MCT4, which control the import and export of lactate, a key molecule in TMVC cells metabolism. In the pathway map on our right, it shows that MCT is in charge of bringing lactate into the cell, and MCT4 is in charge of bringing lactic acid out of the cell. The following slide shows the core metabolic pathway that ACD disrupts. In untreated TNVC cells, lactate isn't just waste. It is actually used as an energy source. Lactate enters the cell through MCT1, is converted to pyruvate by LDH, and then it enters the Krebs cycle. This fuels oxidative phosphorylation, allowing the cell to produce large amounts of ATP efficiently. When ATP is made this way, the cell doesn't need to rely heavily on glycolysis, so glycolytic stress is reduced. In fact, this process actually downregulates glycolysis, helping the cell stay efficient and avoid acid buildup from excess lactic acid production. When ACD blocks MCT1, lactate can no longer enter the cell. This leads to less pyruvate, less ATP, and a compensatory increase in glycolysis, which generates more acid and a drop in the pH. If MCT4 is still functioning, it can export that lactate acid out of the cell. But if MCT4 is blocked or overwhelmed, acid stress builds up and the cell becomes metabolically unstable. That's why researchers, including me, are interested in the dual inhibition of MCT1 and MCT4 as a way to fully shut down this energy recycling system. So based on that pathway, my research question is, if we block MCT1 with ACD, do TMVC cells adapt by increasing MCT4? To test this, I'm using the following three methods. First, I'm gonna use qPCR and Western blotting to measure MCT4 expression over time after ACD treatment. Second, I'll use live cell imaging with GFP tagged MCT4 to see if the protein moves through the cell membrane, which would show that it's being activated for export. Finally, I'll run a colorimetric lactate export assay. If MCT1 is blocked, but lactate export remains steady, it suggests that MCT4 is compensating. And here's what I expect to see. In MCT4, I predict that MCT4 expression will increase over time with ACD treatment, especially compared to untreated and solvent controls. In figure 2, I expect to see MCT4 move from the cytoplasm to the membrane, which would indicate that it's being activated for lactate export. And in figure 3, I'll measure how much lactate is exported under different conditions. If lactate export stays high even when MCT1 is blocked, that would suggest that MCT4 is helping the cell adapt. If that happens, it would suggest the idea that If that happens, it would support the idea that dual inhibition, blocking both MCT1 and MCT4 at the same time, could be more effective than targeting MCT1 alone. This research matters because it shows how adaptable cancer cells can be. By understanding this mechanism, we can better identify which patients are most likely to benefit from AZD based on their MCT1 and MCT4 expression profiles. It also points to the potential of combination therapies, using AZD alongside drugs that inhibit MCT4 to fully block the tumor's metabolic escape routes. More broadly, this highlights how cancer cells use metabolic flexibility to survive treatment, and why targeting these pathways is so important in future therapies. Thanks so much for watching my presentation. I really enjoyed working on this project, and it showed me how a small molecule like AZD can have a big impact if it is used strategically. Feel free to reach out if you have any questions.